So I'll give a brief introduction to one of the two organoid types that we will talk about today. So Sassai, I guess, but also Jürgen's lab with Madeline have championed organoids, essentially exploiting development to build central nervous system systems, or for instance kidney or lung. And in our lab, we came up with a protocol to essentially rebuild crypts of the gut, probably the most rapidly self-renewing tissue of a mammalian body. And just to show the guys who did it, so Nick in my lab found this marker LGR5 that we now believe, we know it's the receptor for aspondin, crucial in the adult stem cell-based organoid cultures. We also think that LGR5 is a marker for each and every active epithelial stem cell, or a cell that acts as an epithelial stem cell, independent of whether it's mesoderm, and ectoderm, or endoderm. So Nick found this a large number of years ago. He's in Singapore at the moment with his own lab. And then when we saw these stem cells in Nick's mice, we realized that they didn't really behave at all like what we had anticipated. So they were constantly dividing, they were abundant, they don't divide symmetrically, etc., etc. And based on this observation, Toshi wanted to take these cells and like ES cells or IPS cells, design a culture system that would allow him to start from one stem cell and make many stem cells. And we knew quite a bit about the growth factor requirements from all sorts of knock-ins and knockouts. So Wnt was crucial, epithelial growth factor was crucial, and from a transgenic mouse that we made a long time ago, we knew we had to block BMP to create a cryptopermissive environment. The expectation was when Toshi would put one of these LGR5 stem cells in this culture medium, we get a lump of cells, stem cells, but much to our surprise, we got these structures that we now know are very good phenocopies of the gut epithelium. So you see crypts, you see villus domains, all the cell types are there, usually in the right ratios. Depending on where you pick them from the gut, they will actually represent exactly that region, so they'll make the hormones of that region, they'll have the transporters of that region, even after a year in culture. So they really are extremely hardwired, fated in a very hardwired fashion, so they're the total opposite of what IPS cells are. You cannot move them out of their fate with growth factors. And we even found, Alexander van Oudenaarde found a few cell types that were previously unknown, first in these organoids. These were hormone-secreting cells, and then when he went back into mice, we discovered that they actually also existed in real life. So they, in the gut, this is probably still the best example. They really seem to be doing exactly what the same tissue does in vivo. So since then, we've come up with lots of other growth factor conditions, and other labs have added protocols as well. And I'll give you three short stories. First one is just out, I think, done by Benedetta Artigiani in my lab, and building on Mary's work about 4 or 5 years ago. So what you see here, and I'm sure Mary will say a lot more about this, is that cholangiocytes, when you put them in culture, almost instantaneously de-differentiate massively. So we think that they probably can all do this, and they become what's probably called by pathologist oval cells. They become bipotent, and you can expand them for a long period of time, and they then have the potential to become cholangiocytes, again, that's where they came from, or hepatocyte-like cells. You then have to transplant them to really turn them into real hepatocytes. And this is what they look like for these very hollow cysts, but they are highly polarized, and I'll show that. And if you make a movie of these cultures, they're pretty dynamic. This probably runs over a few days, but they really are these big balls of hollow structures. So what Benedetta wanted to do is model cholangiocarcinoma in these organoids, and she focused on one particular gene, BAP1, BRCA1 associated protein. It's probably not relevant for its function. There is a fly homolog, which we now believe is the real homolog. It sits in one of these chromatin remodeling complexes that you see here, the deubiquitinates H2A on K119. Mutations of BAP1 are seen, and these are loss of function mutations, in a diversity of human cancers, but particularly cholangiocarcinoma. And there's a lot of literature on what it would do in cancer, and there's a list here that Benedetta extracted from PubMed, and essentially the proposed function of BAP1 is all over the place, but none of these actually relate to the function in Drosophila, where it's a chromatin modifier and nothing else. It's in a polycomplex. So what Benedetta did with CRISPR works very well. So you actually inject all the reagents, so the RNAs, into the lumen of these organoids in a droplet of matrigel. She does a number. She can do 20 or so in a minute. And then she zaps the entire droplet. And you get very efficient transfection of the cells. On the left here you see the cholangiocyte organoids as Mary has devised them, but the moment we hit both genes, both alleles of BAP1, we get these very strange looking structures. You can see them here by H and E, so they're very dense. There's no longer a single lumen. This is the wall of one of Mary's organoids, so this is where the lumen is, this is where the matrigel is, so highly polarized cells, although it's a very thin layer. That is entirely lost. The junctions seem to be lost largely. All epithelial aspects seem to be gone. So this really looks like EMT, and actually better EMT than we've ever seen in the lab. And you can also see this with stainings under the confocal. These are ZO1, so stains at the top of these junctions here. So very nicely polarized normal cholangiocyte organoid. But the moment you remove BAP1, essentially the organization is entirely gone, and ZO1 no longer localizes anything anywhere in particular. And if you then make movies, you remember the balls that I just showed, these Christmas balls? This is what they look like when you knock out BAP1. So they become these slimy, cannibalistic, they eat each other, as you can see, mold-like structures. So very different. So we really think BAP1, you knock out BAP1, and they undergo EMT. Now then, Benedetta collaborated with friends of us in Nijmegen and did a lot of omics. I won't take you through all this detail here, but somewhat surprisingly, if you knock it out, only about 300-400 genes change in expression. So I would have expected thousands for a chromatin modifier, but it's very specific. And when you then ask what these genes are, almost all of them fall into, you can see some names there, into epithelial type genes. Junctional proteins, proteins are involved in polarity. At least 2/3 of the genes fall in that class. So it really looks like BAP1, although it's a chromatin modifier, the ubiquitinase, targets very specifically, at least in this cell type, a program that allows a cell to become to be epithelial. And also, so in the face of these papers, I should show this here, that state all sorts of cytoplasmic functions but no chromatin modifications, Benedetta then re-expressed over 24 hours and linked to GFP a cytoplasmic version of F1. As you can see, nothing much happens. So you see that the GFP and the protein is nicely induced. But if she does the same thing by giving it an extra NLS, so this now goes to the nucleus, in a matter of 24 hours, these organoids totally reorganize themselves back into an epithelial structure. And if you now repeat all these omics experiments, you'll see that at least 250 of the 300 genes immediately go back to the original situation. So again, a very tightly controlled system. So they look very malignant just by knocking out BAP1. However, if you transplant them, they really don't do much. They basically just sit there, the cells. Benedetta then created 4 sort of a quadruple knockout line of these cholangiocarcinomas. They still look pretty normal, they grow pretty normally, but then on top of that she adds BAP1 and we again get this strange EMT phenotype. And if you now transplant these, the ones with the 4 mutations, you can see them here, forms what our pathologists call adenomas, not really malignant tumors. But when you hit, then on top of that you hit BAP1, you go from here to here, and now there's, for instance, this massive fibrotic reaction from the mouse tissue that's apparently instructed by these tumor cells to form these very invasive malignant fibroblastic fields. And these cells will actually metastasize as well. And from this, I think she deduced that the BAP1 tumor suppressor function is really maintaining or breaking this EMT barrier. And I think it's a good example of how you can use organoids, starting from human wild-type organoids with CRISPR, to figure out how exactly a cancer pathway is controlled. You could do this in mice, but it would have cost years, and this project probably only took about a year or so. And then finally, quickly, this is a paper we're trying to publish currently. These are 3 PhD students. Yeah, it took me a while to realize that actually they believe that this looks like a rabbit, and the snake is catching the rabbit. So I don't know why they did this, but they at one point they told me that they had laid their hands on a Cape Coral snake. And in Holland you cannot kill snakes, it's illegal, but you are actually allowed, snake breeders can open snake eggs. And a snake fetus or embryo, what they're called, are not considered to be animals under Dutch law. So that's what they did. And these actually, so this is a few days before this would have been hatched out of the egg, and they actually, at that time, they can hunt. They have eyes, they have venom, they have teeth, and because they have to start feeding themselves immediately. So they have venom glands. First, they realized they could grow liver and pancreas and lung from these snakes under the standard mammalian growth factor conditions. So these apparently bind to all the relevant receptors. And then they started growing these venom glands. And you can see this gray. This is one of these organoids. This gray stuff is the venom. You see that's secreted. You see these vacuoles. They look like penis cells to some extent. These dense secreted bodies. So then they decided, well, let's do an RNA-seq, single-cell sequencing, because you cannot publish papers anymore without single-cell sequencing data. But they were a little worried because we had contamination problems. The mouse intestinal organoids grow so fast, they're like HeLa cells, so they tend to get into other people's cultures. And so they were a little worried because they grew, whether they were snake. And so when they got their first results back, there were 2 surprises. Well, first one, it looked like a snake. The second one is that they then realized that there was no annotated genome, so they couldn't do anything with the data. So it then turns out that actually there is software that if there is a species that remotely resembles your species, you can actually run this software and annotate that genome. So king cobras had been sequenced. And apparently the king cobra is bigger than the Cape Coral snake, but they found the king cobra and then annotated. And what they essentially find, and there's now better plots of this, but they find this whole stem cell hierarchy. There's different cell types that make different families of toxins. They probably make about 20 or 30 heavily modified proteins, so they have to be modified in a very specific way. HEC293 cells cannot do this. There's strange disulfide bridges, strange sugars on these peptides to make them toxic. Actually, there are LGR5 stem cells. MASH2, ASCL2 for the gut aficionados is there. TROI is there. All of the markers that you would see in the gut stem cells in a mouse are expressed by the cells that sit somewhere here. Also, so there's about 3 or 4 toxin families. Also, it wasn't known whether all cells will secrete all toxins or whether they're specialized, and it looks like there's 3 or 4 different lineages that make these different toxins. Now, since then, we befriended for the Dutch here Freek Vonk. And Freek Vonk is the Dutch homologue of this guy, this Australian guy, Irvin, who died, who was killed by a stingray. And he looks the same. So Freek is blonde and a big guy, and he has shark scars in his shoulder. And so he travels the world to wrestle with crocodiles and snakes. And so everywhere he goes, he sends us bits of reptiles. And I'm almost done with my talk. So, so when I first saw this, I told Georg, why do you grow snake venom glands? You could have grown crocodile tear glands. And then he said, well, I'm actually growing human tear glands, and he can make them cry. So he can actually add the right neurotransmitter, and they start producing tears. And so now Freek has committed himself to get us some crocodile tear glands, and see if you can grow and then produce crocodile tears. Anyway, so the venom that's produced is by mass spec, so it's difficult to see how these proteins are modified, but you see the numbers here. This is from when they milk these snakes, the exact molecular weights, and these are the main peaks in the venom that's produced in these organoids. Actually, they produce much more than snakes produce per gram of tissue, so they're extremely potent even after 1 or 2 years. And the molecular weights essentially are identical, so we think that they really are the venom gland producers. And with that, I'll hand over the floor to the next speaker.